the next chapter we're going into, the next unit is really taking Mendel and taking it to the next stage, right? Take it to the next step. As you saw in the video you watched at MIT, Mendel had no clue what chromosomes were, which is pretty amazing because his inheritance pattern, these Punnett squares, that the mathematical model he came up with to pattern inheritance really follows close with how chromosomes sort and how chromosomes align. So it was really amazing that he didn't really know what chromosomes were. So what we're going to shift into, a little bit more complexity of a little bit more complex types of inheritance patterns, and we're going to talk a little bit about chromosomes and their role in inheritance. Pretty straightforward. This is all based off of meiosis. I know you guys did the time-lapse videos, and at some points I will definitely, before the next test, assign who's getting the extra points because they're all very good. But just kind of highlighting, like when we're doing these Punnett squares, really what's going on is meiosis. So we can kind of give a quick breakdown of meiosis here. I will recap it again. I will kind of go over the key things with you for meiosis. But in any case, we kind of see how our round yellow seeds and our green wrinkled seeds, how gametes can form, how alleles will segregate, independent assortment, all that good stuff, in the formation of gametes, with then eventually the formation of new organisms. So just keep in the back of your mind, all we're talking about here, all this rearranging and shuffling, it's all meiosis. That should be in the back of all of our minds. The first person we want to talk about today is Thomas Hunt Morgan. Thomas Hunt Morgan was a scientist. He worked at, he started what was known as the Fly Lab. Does anybody know where Thomas Hunt Morgan did his work? Less than a mile from here, probably less than a half mile from here, at Columbia, right? He did his work at Columbia, pretty close to here. And he studied Drosophila melanogaster, otherwise known as fruit flies. Why did he study fruit flies? He studied fruit flies because they were small. They had quick life cycles. They would turn over real quick. Every couple weeks, you get a new generation, and they would produce large numbers of offspring, fairly large numbers of offspring. Right. So why did he use fruit flies? They were easy to work with. He looked at a number of different traits. He looked at wing types, body color, but the one I'm going to talk about first is eye color. He looked at eye color in fruit flies. And he did something a little bit different with how they did the notation. You'll see this commonly with a lot of different genetics problems. Instead of sticking with the old-fashioned big T, little t, they'll show it a different way, where they show the recessive trait, and then they put a plus next to it for dominance. So this would be tall. Right? So now it's over here is what we're seeing, his crosses. Right? White eyes are recessive, and the W pluses are the dominant color, which is red. So red eyes is dominant over white eyes. Everyone okay? Mo, you can kind of like get this stuff as well too. Okay. All right. So pretty straightforward, right? If we're looking at crossing red eyes and white eyes, right? We do a pretty simple, straightforward cross. We believe that red is dominant over white. I just kind of said that. Here's our cross here. I have a homozygous red-eyed fly crossing with a homozygous white-eyed fly. Homozygous recessive, homozygous dominant. Right? In that first generation, what should we see? 100%? All 100% red eyes. We cross the F1 generation. So we cross two heterozygotes. W plus, W plus. W plus W, W plus W. We get across here, we get a 3-to-1 ratio, right? 75% red eye, 25% white eyes. And that's pretty much what he saw in his lab. But he looked a little bit closer and he noticed something very strange about it. Right? He did get the 75-25 breakdown, but he noticed the white eyes were only present on the male flies. There were no white-eyed females. Kind of weird. So he thought a little bit about the inheritance pattern and said, you know what? There's got to be something else going on here. Obviously, red eyes must be linked to the X chromosome. So we've already kind of talked about color blindness, hemophilia. Right? It was on the quiz today. But so this is just kind of how they got there. Right, all he developed, Thomas Hunt Morgan, really came up with X-linked inheritance. So now let's do the F1 cross again. It's going to be done a little bit different because I'm going to have a female that is heterozygous for eye color. She's a red-eyed female, right? And I'm going to have a male, right? So these would be my F1 generation. Looking here, right, in the F1 generation, they're all red eyes, correct? So they're all red eyes. He's red eyes. She's red eyes, except she happens to be heterozygous. Now when we do the cross, now we see that 25% of the flies with white eyes, but they're also males. So there's nothing new. You actually already knew this. We've already done this already. I just wanted to show you how his data brought them to understand a little bit about X-linked inheritance. So that was Thomas Hunt Morgan in the fly lab right across the street. He had another gentleman working there, an undergrad student, or a graduate student. I forget which one. He might have been a graduate student. Who was working there as well, helping them kind of go through their studies. But before we get there, just a couple quick things. When we talk about fruit flies, we can talk about wild types and mutants. Really, any organism we can talk about. The wild type is what you see normally. So in this case, what do you think the wild type is? The red eyes or the white eyes? The red eyes. The dominant trait's going to show it more. So that would be the wild type, which would make the white eyes the mutant. This is just simply walking you through what we've already done. Just kind of walk you through what we've already done again so you can reference it later. Here's the cross that they did, right? In the F1 generation, we had all red eyes. Right? The female happened to be heterozygous though. When we cross them for the F2 generation, we get that white-eyed male showing up. So again, this is what we just did on the board. Okay, so now, let's get to this other gentleman named Alfred Studevant. He was a student in Thomas Hunt Morgan's lab, and helping him do his work, doing all the little tedious stuff that interns do in laboratories. But he was also had some extra time in his hands, and he liked science. So when you're in a lab, you're constantly collecting data. Remember, when you're experimenting, you don't know what the answer is. Most of the times in a high school setting, you kind of know what the answer should be, and then you try to get your data to fit that. These people have no idea what the answers are. They don't know what the inheritance patterns are. So they get data that have to sort and find patterns, which means sometimes you get data that you can't quite find the pattern. It doesn't really make sense to anybody. And that's what there was. There was a folder of data sitting in the lab that Alfred Studevant said, hey, I know people are trying to figure out what that pattern is. Nobody has. Could I take a look at it? Thomas Hunt Morgan said, yeah, sure. Take it home, kid. Have fun. So he took it home, worked it at night, kind of kept studying it, and he actually started to make some sense out of the data. And that's what we're going to go through now, is kind of what the progression he went through was. Just a couple terms. When I'm doing a cross, and I'm looking at two traits at a time, so it's kind of like a dihybrid cross. If I get flies that look just like one or the other parent, I would call them a parental type. If I get a fly that looks like a cross, has one trait from one parent and another trait from another parent, we would call it a recombinant, because it's like a recombination of the two parents. So just be kind of familiar with those two terms. We're going to use them going forward. Okay, now let's actually mute this for a second and let's talk. So we're going to do a cross here. And we're going to look at body color. We're going to look at body color, and we are going to look at wing type. So this is nothing crazy. They had a heterozygous fly. and I want to find out what something is, I do something known as a test cross. If you want to know what something is, what are you going to cross it with? Something that's heterozygous, something that's homozygous dominant, or something that's homozygous recessive? Homozygous recessive, right? Because if you have something homozygous recessive, what do we know automatically? We know it's, if I have a short pea plant, what do I know about it? I know it must be little t, little t. So if you have something that's homozygous recessive, right, in a dominant recessive situation, you know the genotype. You know the recessive genotype. So he did a test cross, and he's looking at body color and wing type. Okay. Now, the dominant body color, which I'll just kind of note over here. The dominant body color is gray. The recessive is black. The dominant wing type is normal. The recessive wing is vestigial. Vestigial, if you have a vestigial organ, is it operating anymore? No, it's kind of shriveled up, it's gone away. So vestigial wings are wings that are kind of shriveled, they don't really function anymore. So those are the traits he's looking at. And here's the cross. Right, B plus B, VG plus VG, crossed. And we're going to do a test cross here. So we want to really find out what happens with this one. So we're going to cross it with a homozygous recessive, B, B, VG, VG. And we do our cross. If you guys want to note this down as we're going, you guys can kind of jot this down as we're going. I definitely kind of keep track of this for a little bit. So let's do this. Here's our big 16-box cross. We get B plus can match with VG plus, just like we've done a few times. A B plus can match with a VG. A B can match with a VG plus. And a B matches with a VG. Everyone good? Those are gametes we would form from there? B plus, VG plus, B plus, VG, B, VG plus, B, VG. Nothing new. All right, so now we could cross it. We'll cross it here. On the other side, it's pretty easy. They're all the same, right? So they're all B, VG, B, VG, B, VG, B, VG. We get our cross. We're going to get B plus, B, VG plus, VG, B plus, B. Actually, it's going to be the same. Sorry, I meant to go this side. It's going to be the same all the way down. We're going to get B plus B, VG plus VG, same all the way down. B, B, VG plus VG, same all the way down. B, B, VG, VG, and same all the way down. All right, what's our ratio? Yes?
I think this is actually process on the second.
This one?
Yeah.
Should be B plus B, VG, VG, VG. Thank you. Everyone okay with that? All right, what's our ratio? One to one to one to one. Let's pick a random number, 2,300. If I had 2,300 offspring, how many should I have of each? 2,300 divided by 4 is? No, 2,300. Not 23,000. You're close though, 570. Or 575? What is it? 575. So we should end up with 575, 575, 575, 575 as our expected values if we had 2,300 in the population, correct? Okay. So that kind of makes sense, and that's what we would look to see in the data. This is really the data tables that Student was looking at. This is what the expected numbers were, and that's not what they got. Okay. Something else is going on here. So let's kind of take a look and see where we are, because that did not the data comes nowhere near those numbers. If we were to do a chi-square, it would be very, very, very non-significant. It would tell us that is not the inheritance pattern. So, before we go on though, this is what I wanted to say. This group here, is this a recombinant or a parental type? One parent is gray and normal. One parent is black and vestigial. So is this one here a parental type or recombinant? Parental, right? Because it looks like one of the parents. What about this one? That's a recombinant. What about this one? Recombinant. And this one? Parental. Everyone sees parental recombinant, the ones that look like the parents, the ones that are a mix? Okay. All right. So again, we did this whole thing. That's what we expect. That's not what we got. Who can tell me why that's not what we got? What did we just assume with what we just did, and why is it wrong? Oh man, yeah, go ahead, man.
We assume that some of the traits were like dominant, and some of the traits
No, they are dominant recessive.
We assume that they're like not on the same chart.
We assumed what law of Mendel? The law of independent assortments. And can these traits actually assort independent of one another? No, because these traits are what? On the same chromosome. Notice, look at this B. Just look at this one right here for right now. Make it very simple. That B plus, can that B plus match up with that VG? No, that B plus is linked to the VG. That B is linked to that VG. A little different, right? So now what would our Punnett square look like? So now let's mute it again, and let's do what our Punnett square should look like now. So now, we'll actually leave that on. We'll do it over here. So now if I do my Punnett square, knowing a little bit more information, one allele would be B plus VG plus. Could it separate? No, so therefore you're going to get two of them because of the number of gametes we form. And then you're going to get BVG, BVG. Does that make sense? According to this, because these two can't separate, and these two can't separate, so this is technically what our Punnett square should look like. Makes sense. So now we do it, right? BVG, 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 all the way down, all the way down, all the way down. Right, we get our cross, B plus VG plus, I'm sorry, B plus B, VG plus VG, B plus B, VG plus VG, B, B, VG, VG, B, B, VG, VG. What type went away? Which one? The recombinant went away because they can't assort independently. So if one parent is this, those dominant alleles are going to stay together. So in this case, we really only get a 1-to-1 ratio, right? Because we only get two types. We get gray normal, so these are parental dominants, or we get parental recessives, black vestigial. Everybody see that? So now between that one and that one of them is going to be supported by the data. Okay, so if we had 2,300, how many do we get for each of these? You'd get, what, 1150 of each? I can't do math in my head. About 1150 of each. Okay, you want to see what they really got? This is what they got. He went off the Punnett square we just did initially, independent assortment. So he came up with the 2,300 of a 1-to-1 to 1-to-1 ratio. Right? 575, 575, 575, 575. However, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, 575, there's something going on because this actually is kind of on the right ballpark as well with 1150, 1150. However, when he got his data, neither one were right. They got 965, 944 of the parental types. So the parental types showed up a ton more. And then the 206 and the 185 were the recombinant types that showed up every so often. What's going on? What is it? Crossing over is going on, right? Remember, alleles, the alleles are linked to one another, but what you did in prophase 1 when you did meiosis, you did crossing over. And if the alleles are cut away from each other, they can separate. So although they are linked genes, linked alleles on the same chromosome, if crossing over occurs, they can be separated from one another. Does that make sense to everybody? So really, neither of those Punnett squares are correct. They're both kind of going around, beating around the bush. But what Alfred Studevant came up is he came up with the concept of crossing over, and he came up with something known as a linkage map. So this is just kind of focusing in on the data here, and what he calculated something known as a recombination frequency. Recombination frequency is pretty easy, right? Where did he get the 391 from? The sum of what two? Recombinants, right? You just count up how many recombinants we have. 206, 185, divided by our total number, 965, 944, 206, 185 equals 2300. That's the number we've been going over. And that gives you a recombination frequency of 17%. So what that's saying is, let me actually erase real quick so we have some space here. What's that saying is, if I look at these genes right here, it means that crossing over occurs between them about what percentage of the time? About 17% of the time. All right, I'm gonna give you another piece of information. I'm gonna give you an eye color. An eye color, let's say it is red eye, so W plus. All right, and I'm gonna say the recombination frequency between the eye color and body color equals 7%. And the recombination frequency between eye color and wing type equals 10%. What is that telling us now? So all I'm doing here, we did the experiment we did it all through with the body color and wing type, and we got a recombination frequency of 17%. I redo the whole experiment again, except now I compare the eye color with body color. And I get a recombination frequency of 7%. And I do the whole thing again, and I get a recombination frequency between eye color and body color of 10%. What is that telling us about the location of the eye color gene? Here's my large chromosome now. Right? Here's my B plus. Here's my VG plus. What's the space here related to? What's the recombination frequency?
It's between 7% and 17% in between.
Well, what's this number though? What's it related to?
10%.
Relates to 17%. That's a recombination frequency. Because of this spacing, it recombinates 17% of the time. Now, if my eye color and body color is 7%, where does that fall on the line? How about somewhere right about here? It's telling us about spacing. And this one here 7%, 7%. It's telling us about the spacing between the three. So what recombination frequencies are telling us, you know, I'll go with just looking at recombinant types, but the larger the recombination frequency is, the further apart the genes are on the chromosome. More likely that crossing over occurs between them. The closer the genes are together, the less likely crossing over is going to occur between them, the closer they are together. All right, so just kind of finishing up with recombination frequencies. Get you out of my scribbles. So what we just made here, when we kind of put the genes on the chromosome, we made what's known as a linkage map, kind of linking our genes together and mapping out the distances between them. And this is basically what we just did. I have 3 different genes. I do my experiments. I get a recombination frequency between B and VG, and that recombination frequency works out to be about 17%. I then do my experiments again between the CN. CN is an eye color known as cinnabar. Between the CN and VG, I get a combination frequency of about 9.5%. And then I get a recombination frequency between CN and B about 9%. What do you notice about the 9.5 and the 9 compared to the 17? They don't add up together, right? Because remember, this is all based off of experimental data. You're not going to get exact, precise numbers. It's going to be off a little bit. But by looking at our recombination frequencies, we create what's known as a linkage map. Every frequency, every percentage point is 1 unit on our map. So if I get a recombination frequency of 1%, it's telling you that those genes are really, really, really close together. It means only 1% of the time does crossing over occur between them. The max recombination number is actually 50%. So if they're as far as they can be apart, recombination maxes out at 50%. In fact, Mendel did his experiment, and 2 of his traits, I forget which 2, I'll look it up for you, 2 of his traits actually were on the same chromosome. But it still supported dominant recessive because they were so far apart that they were separated, recombinated almost all the time, or 50% of the time. So it didn't really affect his data. All right, you can stop.